Zooming in on this high-res image of a mouse brain reveals rectangular lines where images were stitched together, with each colored dot representing a brain cell type. The task of understanding the inner workings of the brain has fascinated both philosophers and scientists for centuries. Santiago Ramón y Cajal's neuron doctrine launched a race to understand the cellular composition of the brain and how brain cells are connected to each another. A century since, modern tools called neurotechniques have given neuroscientists a way to closely inspect every component of the brain. By mapping out how all the cell types in the brain are organized and examining how they communicate with one another, neuroscientists can better understand how brains normally work, and what happens when certain cell parts go missing or malfunction. To deconstruct a thinking machine made of billions of neurons, we need a parts list, an inventory of the brain's cell types. The task of understanding the inner workings of the brain has fascinated both philosophers and scientists for centuries. Aristotle proposed that the brain is where spirit resides. Leonardo da Vinci drew anatomical depictions of the brain with wax embedding. And Santiago Ramón y Cajal, with his 1906 Nobel Prize-winning work on the cellular structure of the nervous system, made one of the first breakthroughs that led to modern neuroscience as we know it. Using a new way to visualize individual cells called Golgi staining, a method pioneered by Nobel co-winner Camillo Golgi, and microscopic examination of brain tissue, Cajal established the neuron doctrine. This principle states that neurons, among the main types of brain cells, communicate with one another via the gaps between them called synapses. These findings launched a race to understand the cellular composition of the brain and how brain cells are connected to one another. Jumping forward 100 years to today, modern tools called neurotechniques, which include brain mapping, have given neuroscientists a way to closely inspect every component of the brain. The process is like finding a needle in a haystack. It would be a lot easier to find if the needle, or cell type, glowed. This can be done with either genetic or immunostaining methods. The genetic method takes advantage of animals, like mice, that can be genetically engineered so only the target cell type is visible under specific fluorescent lights. And through several steps, once scientists are able to detect their target cell type in an image dataset, the final step is to locate specific cell features in a reference brain. This reference brain serves as a standardized map that shows where each brain region is located. Scientists can then use this map to compare with individual brains and note their variations. These steps are repeated for each cell type, creating a richer and more complete map of the brain with each run-through.